A large number of transcripts from the sweet potato root are involved in the process of tuberous root formation and development. To identify these transcripts, we generated thousands of sequences using Illumina paired-end sequencing, and many were annotated with ontology and mapped to KEGG pathways. Several cDNA SSRs, cSSRs, were identified as potential molecular markers in our unigenes. After amplification of 92 primer pairs, a high-throughput sequencing approach was used to validate the genomic sequences. The result of the present study will help to discover novel genes and to develop potential molecular markers for sweet potato breeding.
This article is titled De Novo Assembly and Characterization of Root Transcriptome Using Illumina Paired-End Sequencing and Development of cSSR Markers in Sweet Potato, Ipomoea Batatas, and was authored by Huang Lifei, Luo Zhongxia, Zhang Xiongjian, Chen Jingyi, Fang Boping, Wang Zhongying, Chen Xinliang, and others.
We are article.tv. Article links can be found in the video description.